IDEX, we think this internal alert system is a very good system, personally. It is not too expensive and not too time-consuming, and actually it's really easy to analyze. So anybody could do the analysis of these samples, and that's one of the reasons that we chose it. We also chose it because Riverkeeper also does the same thing, so our data is comparable to Riverkeeper's, which is really important to us. The things that you would need, especially since almost all of our sites that we sample of are in brackish water, is obviously you need your sample. We're actually going to only use 10 milliliters of our sample. And then we get these little sterile water bottles. We don't have the facilities here to make our own sterile water. So we use 90 milliliters of this and 10 milliliters of our sample and put it in our mixing bottle. And then put in our reagents. And all this reagent is going to do is when you mix everything together, it's basically food for the bacteria. We're looking for 2 Enterococcus species. And when basically they eat the reagent, it puts a fluorescence tag on their DNA. So after incubation for 24 hours, which you incubate it in this, is called a quantitray. And you're just going to compare your fluorescing small wells to large wells. And you'll use something called an MPN chart, which if you guys want to try later, you can get up close and like touch my samples that I already took in the back. And so after incubation for 24 hours, you pull these out, you go into a dark room, you're going to use a UV light, and if there's any bacteria in the wells, they will fluoresce. And then IDEX and the EPA actually created this chart, and it's an MPN chart, which stands for most probable number. And so you'll be getting the most probable number of bacterial colonies per 100 milliliters of sample is what you'll be getting when you do this sort of test. And actually, because you're only taking 10 milliliters of sample, because we are in brackish water, you're going to move the decimal point over to the test, and this test allows for greater than 24,000 bacterial colonies, which a lot of other tests that are simple will only go up to 100 bacterial colonies per 100 milliliters of water. So that's another reason that we chose this system, is because it can get higher, more accurate numbers for what you're doing. Especially, our program is geared more towards boating sites. So that's more a secondary contact, so you don't have to be as careful with your numbers as with swimmers. You might be more cautious. Alright, so if we head into
are they bacteria concerned, like E. coli or?
We only do Enterococcus, and this is an indicator bacteria. So it basically needs the same exact things that some of the more harmful bacteria would need. And we are looking for 2 species of it. And so, like if I spilled something on myself in the lab, I wouldn't have to worry, but it is an indicator and approved by the EPA for how much other harmful species might be in the water, and a good indicator for how much sewage is actually flowing into the water.